Hey everyone, welcome back. In this video, we are going to be talking about proteins and how the topic of proteins gets spiraled throughout the entire year. So in the different units, we always brought up the topic of proteins. In Unit 1, we're going to talk about the chemical structure and the properties of proteins. In Unit 2, we're going to talk about how proteins are made and the role that they play in the fluid mosaic model. In Unit 3, we're going to go into a specific type of protein, the enzymes, and their role in the biochemical pathways such as photosynthesis and cellular respiration. In Unit 4, we're going to talk about the role of proteins in cell division, in regulating the cell division, and how they help the process of cytokinesis. We're also going to talk about how genes that code for proteins are inherited. Then in Unit 5, we're going to talk about the role of proteins in DNA replication and organization. And we're going to talk about gene expression, which is how the proteins are made, and the regulation of gene expression. In Unit 6, we're going to be looking at how we use proteins to build phylogenetic trees and how variations in the proteins are subject to natural selection. So in Unit 1, when we started the year, we talked about the chemical structure of proteins. They're made of carbon, hydrogen, oxygen, and nitrogen, and the bonds that form between them are considered peptide bonds. We also talked about the monomer of the amino acid as an important example of how structure affects function. So let's take a closer look at the structure of this amino acid. First, we have a central carbon, and recall that carbon can form up to 4 pairs because of the 4 valence electrons. Now, all amino acids have a common structure of a carboxyl group, an amine group, and a lone hydrogen. Now, the R group is what makes each of the 20 amino acids different from each other. The R group can be either classified as hydrophobic or hydrophilic based on the charge of the side groups. It can also be considered large or small or sometimes even aromatic. Now, you are not required to memorize all 20 amino acids, but you are required to understand that the R group will determine the overall function of that amino acid in terms of its polarity and its ability to interact with the aqueous environment or with the rest of the chain of the protein. Another good example of how structure affects function is the protein folding. So keep in mind that protein levels of folding must be correct in order for the protein to function properly. Now, the 4 levels of protein folding are the primary structure, which is the specific amino acid sequence determined by the DNA itself. Secondary level of protein folding, which is an alpha helix or beta pleat, which will be determined by hydrogen bonding forming between the amino acids themselves. The tertiary level of protein folding include the hydrogen bonding that it forms, as well as hydrophobic interactions. And the main thing about the tertiary level of protein folding is that you want to minimize those hydrophobic R groups from the aqueous environment inside the cell. The quaternary level of protein folding is what's called the globular structure, and that's where you have multiple chains coming together and interacting to form one functional protein. The last thing I want to talk about for Unit 1 is that in the amino acids, it's that R group that determines those properties. So again, you are not required to memorize which ones are which. You are just required to understand that some of them are going to have different shapes, different polarities, and that polarity is going to play an important role in the structure of the protein as it interacts with the plasma membrane, and then the size of the protein is also, or the amino acid is also going to affect how for example, the active site in an enzyme is going to be able to fit the specified substrate for that enzyme. In Unit 2, Cell Biology, we're going to first start talking about how proteins are made. So proteins are made in the ribosomes, and we'll get into more detail about the structure of those ribosomes in Unit 5. But keep in mind that for both prokaryotic and eukaryotic cells, there are ribosomes which are not membrane bound, found in the cytoplasm. In the case of eukaryotic cells, some of those ribosomes are also going to be found embedded within the rough ER. Now, it's helpful to remember that the ribosomes that are embedded in the rough ER are the ones that are going to code for proteins that normally either get secreted out of the cell or that are going to form protein channels that are embedded in the membrane. Proteins also play an important role in the cytoskeleton of the cell. Each of the types of proteins that make up the cytoskeleton are different sizes. You have, for example, actin, microtubules, and intermediate filaments. And all of these make up the cytoskeleton piece and vary in their function and their size. Now, the big thing in the second part of cell biology was when we talked about proteins acting as channels in the membrane. Keep in mind that with facilitated diffusion, no cellular energy is required by the cell, but since the molecule cannot pass through the membrane due to its polarity, it's going to have to go through a special channel in order to go down its concentration gradient. And the example we talked about in class was aquaporin, which would allow water, which is normally polar, to cross the membrane. Proteins also play an important role in active transport, such as what we saw with the sodium-potassium pump. Because sodium and potassium are both polar, they would not be able to travel through the membrane on their own because of those hydrophobic tails. But in the case of active transport, extracellular energy is required, and that extra energy is going to be in the form of ATP because the molecules are moving against their concentration gradient. In Unit 3, we talked about the role of enzymes or a specific type of protein in the form of enzymes. And remember that enzymes are protein. They are organic catalysts. They increase the rate of a reaction by lowering the activation energy, and those enzymes have specific sites for their substrates. Now, the sites of the substrates are called the active site. And the shape and polarity of the active site is very important to make sure that the substrate is going to be able to interact with it. This is where protein folding becomes very evident. Now, keep in mind there are factors that are going to affect the rate of reaction. We use the term denaturing for when an enzyme is no longer able to function. Now, this denaturing process is going to be because bonds are broken and the structure of the enzyme is no longer correct. Factors that are going to denature enzymes include pHs that are outside their optimum, temperatures, ion concentrations. But other factors may affect rate of reaction as well, such as cofactors and coenzymes, which will help reactions along. Enzymes are a critical part of biochemical pathways. They regulate and affect the pathway rate. They can be controlled, these pathways can be controlled by allosteric inhibition, which is where a site other than the active site is affected by a molecule. Cell respiration and photosynthesis are both good examples of biochemical pathways that are regulated by these enzymes, and they use ATP synthase to generate ATP either through oxidative phosphorylation or photophosphorylation. You don't have to memorize the names of the enzymes involved in these pathways other than ATP synthase. But you definitely need to know that these are typical biochemical pathways. They can be entered at any point in the pathway, and they are controlled by enzymes. In Unit 4, we talked about CDK, which is your cyclin-dependent kinase, and cyclin, which are both types of proteins. So keep in mind that your CDK levels remain constant throughout the cell cycle, but the levels of cyclin are fluctuate because cyclin is being produced or being translated from the DNA. When the cyclin and the CDK combine, it forms what is known as the mitosis promoting factor, which will trigger the next stage of the cell cycle. Proteins are also involved in cell division because proteins make up the microtubules. The microtubules were going to form the spindle apparatus, and it's the spindle apparatus, or these microtubules, that display dynamic instability that are going to align the chromosomes up during metaphase and then separate the chromosomes during anaphase. Proteins also have some structural roles that we talked about in this unit. They are, as I mentioned, they are involved in the cytokinesis process as well in form of actin, which will form the cleavage furrow in animal cells as they are undergoing cytokinesis. And proteins are also involved with forming the tetrads that form during crossing over, that allow those homologous chromosomes to exchange genetic information. Unit 5 was a big unit for proteins. We started off by talking about proteins involved in DNA replication and in structural components. So in DNA replication, the enzymes involved are the ones that you do have to memorize and include your DNA polymerases 1 and 3, ligases, helicases, and those topoisomerases. In terms of structural support, proteins were shown as examples of your single-stranded binding proteins and the histones that the DNA wraps around. For gene expression, which is the production of the proteins, this process is divided into 2 phases. Your transcription phase, which first uses DNA as a template for mRNA, and then keep in mind that that mRNA is modified before it exits the nucleus in eukaryotic cell. Some of those modifications include the removal of introns and splicing together of exons by spliceosomes, as well as adding caps on the 3' and the 5' end. Then the immature mRNA is ready to be translated, and that is where that mRNA is used to create proteins in the ribosomes. Now, the ribosomes themselves have the large and the small subunit, and the large subunit is broken up into the E, P, and A site. And what happens is the mRNA is read in these series of triplets called codons, which then code for a specific amino acid. You will always be given the codon chart on the AP exam. The last part of Unit 5 was controlling gene expression. And there were several ways that this can happen. So you can control the amount of protein that's being made by either altering the DNA sequence to either increase or decrease transcription. This would be, for example, as methylation or acetylation. You could alter the protein folding itself, and this is done in the Golgi apparatus, which modifies proteins. You could use transcription factors to increase the protein production, or you could use RNA interference, which are molecules that destroy the mRNA, the mature mRNA in the cytoplasm, to turn off protein production. Now, in prokaryotic cells, there's also the operons, inducible or repressible operons that would control the amount of protein being made to ensure that proteins are made only under specified conditions. But operons are unique to prokaryotic cells. In Unit 6, we talked about proteins can be used to create phylogenetic trees. And the important relationship to understand is that the more similar the proteins are, the more closer the organisms are going to be related. Another piece of evidence of evolution from a common ancestor that we can use is the fact that all organisms on Earth, both prokaryotic cells and eukaryotic cells, use the same exact genetic code. They use the same mRNA and codons to encode for the same amino acid. So this must have been present in LUCA and passed on to its descendants. Last, variations in proteins will produce different phenotypes within the same species. And sometimes those phenotypes will be favored by natural selection and some are not favored by natural selection. So one of Darwin's postulates is that there's variation within a species. So for example, humans might have blue eyes versus green eyes versus brown eyes. So there's variation within the color. And what's going to happen is that some of those phenotypes within a species are going to be favored by natural selection, some are not going to be favored, or some are going to be what we call effectively neutral. Natural selection doesn't work on them or doesn't act upon that particular phenotype. But that phenotype is determined by the proteins that are produced. I hope this video was helpful. Thanks so much for watching. Bye.